No other trust in truth. Hello, everyone. Welcome to Examining Evolution. Today, I'm here with Dr. Dimiter Kunnev, who has a PhD in microbiology. He's done a lot of work in the area of origin of life. And so he's published several articles, and we're going to discuss those today. And really, what's happening here is Dr. Kunnev approached me when he saw my video about the origin of life, and he wanted to present a contrary position. He believes that life evolved by natural processes. And so, of course, that differs with my position, which is we have no explanation for the origin of life, and that it is so implausible to be really an impossible scenario. So, Dr. Kunnev is here today to show me why I'm wrong. So, welcome, Dr. Kunnev. Do you want to say a little more about yourself?
Yeah, first of all, you're wrong, of course. But let's say something about myself. You said microbiology. Actually, it's a molecular biology.
Oh, molecular biology. I'm sorry about that.
And I was studying molecular biology since I remember myself. The difference between molecular biology and microbiology, for the auditorium, they should know that. Microbiology study microorganisms. This is microbiology. Molecular biology study the process of information in the living systems. So, meaning replication, translation, and transcription, and all of the links, interactions, and regulations of those processes is molecular biology. Of course, that includes all the mechanisms in the life, in the living organisms. But major those three components, which we consider today is a central dogma, molecular biology. So how the information is transferred in different ways. DNA from RNA going to proteins and vice versa. So origin of life usually in my student years was kind of interesting area. I never thought that will be area that I will be involved into relatively deeply. However, later on, when I was in some debates with another creationist, especially when I was looking Stephen Mayer's talks, he's completely wrong, of course, Stephen Mayer. In debate, that's happened actually probably 6 years ago, something like that. In the debate, the argument was that information cannot occur, there is no way to get information, those kinds of things, which I'll show you today that it's not the case. But I didn't want to continue to debate at that point. It simply was useless. So I stopped debating. However, in my mind, the thought continued. And I find myself kind of trapped in my own thinking about origin of life, basically. And that's triggering me not constantly thinking and kind of try to figure it out from the molecular biology point of view, not just whoever wrote something. I didn't care at that point who wrote and whatever was written for the origin of life at that moment. So slowly, gradually, within 1-2 years of thinking, I came up with a very good idea and concept for origin of life. And of course, my expectation was that when I opened 2 or 3 papers, which the modern people have, the scientists already figured it out, most of the stuff that I was thinking, Uh, well, it happened that when I started to open now I started to read for Origin of Life and the mainstream scientists, what they're thinking about. And I was shocked to see that whatever I was seeing about, it was not at all presented from the mainstream science. And I said, oh my goodness, what to do? I mean, I have an idea here which it's could explain pretty much everything about origin of life. And at the same time, nobody's presented it already. So I trapped, I was feel trapped in my own kind of thinking about all of my ideas. So I was listening to the radio one morning, and the radio was with the music. I might have only one match, but I can make an explosion. You know this song, right?
I don't know it, but
It was from the Platinum, so it's a very famous song. So
okay.
I said, okay, so obviously that's the case. I have only one match, but I might make an explosion. Who knows? So, and of course, with friend of mine, I continue. He's in Bulgaria. Uh, Together, we published the first paper in 2018, and after that, another paper 2020, and the third paper 2021. Actually, the last paper, which is the origin of life, the point of no return, is the the first paper that was in my mind.
Mm.
So, but the things didn't go exactly as I wanted to be. So I published first the hypothesis. After that, the following from my hypothesis comes the origin of the genetic code automatically. It's like a key. If you have key, it's open the door. You open the door, you have another door, but you already have a key. So immediately you find the key for next door, and everything falls in exactly start to make sense. And now, right now, I'm working on a fourth paper, which is more like a mathematical model and more like a simulation. Unfortunately, I cannot do that in the lab. That is the condition. So at least it's best to do some sort of simulation and to see proof of principle how it might work. Things.
Okay,
So let
so
me, yeah,
you feel
yeah.
like there is an explanation for the origin of life, and that's what you're presenting.
Oh yeah, absolutely. There is a very very clear cut. Because right now I'll tell you first of all what is life, because people don't know what is life. Now I know what is life. Uh, I mean, from the perspective of, let's say, workable perspective of the science, what is life, like a system. This is not philosophical answer of what is life. Everybody have its own explanation, what is life from the philosophical point of view. I'm talking more like a practical. So, for example, if you go in another planet and you find some kind of interactions between polymers, and you don't know, okay, these interactions between the polymers, is it going to be in the road to form a life, or they are already life, or they are not life? Those kinds of answers I can tell you. So before that, briefly, I will describe what is the current situation in the science for origin of life. Of course, it's briefly, there are so many, approximately, there is at least 2,000 significant papers at the moment for origin of life, at least, which one third of the papers are prebiotic. There is a huge portion of this paper which is just discussion and ideas. And there is another one which is the so-called top-bottom approach. Uh, and I'll tell you a little bit about that. So it's a huge field, and the scientists don't agree many times each other, and they agree also many times each other. So the fact that the scientists very frequently do not agree each other, that is very easy bait for creationists to try and to attack idea for origin of life. But I can tell you that doesn't make any good for making a point about intelligent designer, because that's how the science works. The science have a hypothesis. Scientists have different ideas and they might collide each other. They might be controversial each other. But that's not the problem for the science in the long run, because after all, someone will do the right experiment and will show what is right and what is not. So also there is several extra issues outside of just having a scientific process. For example, the funding in origin of life is very low. We don't have money to work on origin of life. After all, that's not a cancer research. And who cares how origin of life was from the perspective of the big money makers? So there's only probably maybe about 5 laboratories right now which are actually, in fact, doing experiments for origin of life. Mm-hmm. All of the other don't have money, and unfortunately, the science is a very expensive field, even in origin of life. I wish to have access to good mass spectrometer to make a very good experimental, to do good experiments, but I don't have access to mass spectrometer. But that's a different issue. So we are not here for that purpose. So there is several kind of mainstream view for origin of life. The oldest view is so-called RNA world for origin of life, and a very long time, probably 30 years, since 1986, when Gilbert created this terminology of RNA world, was kind of so famous that the mainstream scientists usually equalize the origin of life and RNA world, which unfortunately I don't think that is the case now. To me, RNA world is a good try. It's already, to me, this hypothesis, it's already in the history. It was a nice try, but it's not the answer for origin of life. However, that didn't stop all of the creationists to attack origin of life based on the fact that there is a lot of problems with RNA world. And that's a huge issue, because once we do not agree, the scientists do not agree, the creationists immediately catch that and they start to say, oh, I told you, it's not possible. Oh, no. No, that's not the case.
Simply, well, do you feel like you've shown how it is possible?
Yes. Let's keep going faster, because, okay, so I have to be much faster in that case. So yes. The next mainstream view, it's so-called metabolism first mainstream view, which is, okay, before going in metabolism, I have to say a few things. So the RNA world just assumed that there was only RNA entity which self-replicated, and based on this RNA entity, ribozymes, which is enzymes, but only made by RNA. There's no proteins there. And based on this argument, ribozymes trying to mutually benefit each other, and after that, they started to replicate itself, creating entirely RNA entity, which was called RNA world. And from that point, the origin of life continue with so-called polymer transition, where you have to have translation and going in making peptides and proteins. So this is the very short for the RNA world. The next mainstream is so-called metabolism first, mainstream ideas, which it says, okay, there was a reactions which resemble lots of prebiotic and today's building blocks of of the life and also metabolic processes. And those reactions was kind of going in the way of autocatalytic functions, and those was very relatively stable, and from those particular autocatalytic interactions of the like metabolism like processes, life emerged. So that was the metabolism first hypothesis, which I also do not agree. See, that's another way I, as a scientist, do not agree with other scientists. So I don't think
I
this
saw
is the
in your
answer.
paper that you what you're suggesting is that there had to be the translation and the information there simultaneously.
Exactly. So I'll provide you the law for origin of life. There is a law which you have to follow. If you follow the law for origin of life, you can figure it out. If you don't follow the law, you will not be able to figure it out.
Okay,
That's
what is
why.
the law?
So I'll just give me 1 second to finish this particular section. So, and there's a third mainstream of origin of life, which is RNA peptide world. And this is what I belong to, the RNA peptide world, which says you must have a mutual benefit between the short peptides and short RNA, which interact with each other and create a stabilization processes. Once it's stable, these processes, you create Darwinian evolution, or to say so-called information-dependent selection. So once you have information-dependent selection, you can go and jump. And that's happened by mutual benefit between very short RNA and very short peptides. I'm emphasizing very short, not long, short.
Mm-hmm.
What does it mean, short? Mean that it's probable. It's likely to happen. That means short. Okay, so let's go and deep in the RNA peptide world, because right now I cannot talk for everyone and everything, and try to present the main idea and the concept. First of all, usually, especially creationists or Stephen Mayer constantly try to argue that there is no possibility for information to come up, to appear, which is completely wrong. Now, what is information?
What scientific evidence do we have that that's completely wrong?
Let me show you. What is information? Do you have any idea about what is information? Can you tell me what actually is information? Do you have any
I would
concept?
say something that conveys a message, right? Something that conveys a truth about the world, something that conveys a message.
True, but not kind of. So I hope you will agree with me. Information is some kind of pattern of something which have a function, describe or possess some sort of function. That is information. So if you have something which is pattern, and this pattern do something, have meaning of something, it's information. Of course, that's just too overview, because its information could be so many different things. So, for example, starting from something which everybody knows, like writing letter, you have pattern of letters, which means something for humans. That's information, because you have function. of that.
Yeah, let me just read the dictionary definition that I pulled up. Information, what is conveyed or represented by a particular arrangement or sequence of things.
Yep, that's a pattern of something. So you have a sequence or pattern of something. So it is information. Now,
That's nice. Top Dog put it there for us.
Yep. So, however, in the chemical world, information could be just a specific structure. But this structure or specific pattern of something, in our case, information is just a sequence of RNA or DNA in biological world. And of course, this is just one level of information. In biological world, there is many different levels of information. But the bottom one, the very first one, is the sequence of RNA and DNA. You have information also in the structure of proteins. That's also information that possess a different function. Now, starting from that point of view, if you have, for example, let me say a very short oligonucleotides, like let me say 10 base pairs nucleotide. And if you have one of these I'm sorry, not nucleotide, 10 base pairs nucleic acid with around 10 nucleotides. So just misrepresent. So if you have a short oligo, and let's say one of the oligo is entirely based on GC, so GC rich oligo, and another is just AU rich oligo. If you have these two oligos, so you see you have different pattern of nucleotides. One is AU nucleotides, adenine uracil nucleotides for RNA, for example, and the other is GC nucleotide. And you put these two nucleotides, although it's the same size, they will behave slightly differently in the same temperature. For example, the GC rich will melt in a higher temperature because it's more hydrogen bonds containing this double-strand RNA, for example. So you need more energy to put into in order to melt or separate different strands. The AU rich, it have only two bonds, hydrogen bonds, only two hydrogen bonds. And if you put this in the same temperature, or it requires lower temperature in order to melt this particular oligo. This is just a very small example to show how the pattern makes a different function in that case. So in RNA, short RNA, you may have random GC sequences, and because of the different selection forces, the actual stability of this RNA could be different. Not to mention that specifically for RNA, there is another 2D and 3D structures, which also depends on the sequence of the RNA and makes small hairpins and interactions, which is very interesting for the origin of life. And that's also possess a different kind of features, different stability forces for the RNA. So a very small example to show you that actually there is information there. Now, let me show you, so I have to share my screen. Just very briefly, I have to speed up, basically. Let's speed up. Let's see. I can share here.
Thank you for those who have joined us today. Thanks for being here, Otangelo, Top Dog, Doki. Glad you guys are watching. Thanks for your comments and your questions. And Spam. I will get to questions later, and there's some good ones in the chat. Thanks for being here, Mike. And so I hope this is beneficial for everyone. So here's, all right, I've got your
You see it?
screen up.
Yeah. So
Yes.
this is what everybody should know. It's so simple that I even don't need too much of description. But if you have a source of information, and if this source of information determined some sort of structure and function, and this function and structure support the information, so you have basically feedback over the information, that is the condition for Darwinian evolution. Because once it goes in an environment, because of the structure, you might create a situation where you have a more stable form. But the reason to be more stable is because the information containing value or sequence produce this more stable form. Therefore, anything that is changeable in information will reflect the structure, basically. So, and this is what is the definition for Darwinian evolution, which is dynamically stable system where information determines its own supporting structure. It's very simple. Now, you cannot do anything else. You cannot create and upgrade anything in the primordial life form without having such a structure. Therefore, everything that we call life today is exactly possessing this particular structure. That's why this is the definition for life as well. So the life is a dynamically stable system where an information determines its own supporting structure. So also the law for origin of life. During origin of life, the information that I described, the structure I described, and also the rule between them are locked in their origin, meaning you cannot have a process of origin of life where separately you'll have information and separately the structure will appear and suddenly they will merge together and make a life. That's not going to happen. You have to have a own process which describes all of these particular ways. So
okay, wait.
yeah.
There's something I want to stop with because you put up the Terra Darwinia, so I'm going to that part of your article, Origin of Life, Point of No Return. Page 14, it says, in fact, this first life form, we can name it Terra Darwinia. So, and you're saying it's the entity that carried out the first primitive translation event. Okay, and then in the very next paragraph, you say the very steps toward the formation of primitive translation and primitive enzymatic replication are not experimentally demonstrated yet. So what you've said in your own article is that you've invented this first form of life, but there is no experimental evidence. There's nothing in here that actually that we can lock onto and say this is we have scientific reasons to believe in this. And what I noticed throughout all of your articles, Dr. Kunnev, is that there's lots of words about imagining, like imagine a scenario, or it could have been this way, it might have been this way. And so there's really, it's scientific, it's storytelling, but with a bunch of scientific jargon. So what I'm wondering is, where is your actual evidence that this actually occurred?
Okay. The fact that something is hypothetical doesn't make your concept or anyone's concept for intelligent designer in a better situation at all. The fact that something is made, it is kind of you put a hypothesis how something could occur. But don't forget, to make a hypothesis, scientific hypothesis, it's not fantasy. Scientific hypothesis always is based on already proven steps. So if I show you exactly how, according to RNA peptide world, those things happen, you will see that every single step, for example, the interaction between the short peptides and RNA, it's already proven millions of times. I mean, not millions, but there's a significant number of papers which shows that peptides containing arginine, for example, interacts with RNA very well. Not to mention, there is a good papers also that shows that those kinds of interactions stabilize the structure. Inside of the entire hypothesis, you have all the steps which is already proven scientifically. For example, electrostatic charges, interaction between positive and negative ends, interaction and trying to process like aminoacylation on RNA, it's already experimentally proven. So to make from several proven mainstream one concept, which is scientific hypothesis, that doesn't mean you're fantasizing about how could happen. You possess ability to predict something how might happen based on lots of experiment. And I know that creationists are very easy to catch something like we can imagine, oh, we hypothesized. That's not make your concept much better. Simply because, as I told you, scientific hypothesis, it's not an imaginary fantasy. It is based on already proven facts. To make something like to create a hypothesis, scientific hypothesis, like whatever I presented as an RNA world, and I didn't present it yet in front of you, you are sitting on the basis of science, of already proven thing. And just the fact that it's possible in this way, by itself, it's completely eliminate the intelligent designer at all.
Well,
Just
wait
the
a
fact that
minute,
it's
Dimiter.
possible.
Hold on. I think you're familiar with this book because you referred to a quote from it from Dr. Davies and Sarah Murray Walker. You referred to it in one of your articles. So I think you're familiar with it, right? And this from Matter to Life is, about 30 origin of life researchers that their papers have been compiled into this book. And so while you're making it sound like, hey, we've scientifically demonstrated how all these things could happen, well, they disagree with you in this book. This is secular. This is not creationists. Let me read a few quotes from this. And this is from Walker and Davies. The emergence of life and mind from non-living chemical systems remains one of the great outstanding problems of science. And so over and over again, they state that we still don't know. Not only do we don't know how this could have happened, in actuality, what we need is to defy the laws of physics in order to get the origin of life to happen. And we still don't know how it could have happened. So, I have, about 30 secular origin of life researchers that disagree with the claim that you're making here. And I know you're also probably familiar with Dr. James Tour. And one of the questions that Doki asked earlier was, have you seen his abiogenesis video series?
Uh, Probably not all of it, but I'm familiar with James story.
Okay, so James Tour is showing the difficulty. It's not a simple process of life coming together. Another source for this is Dr. Change Laura Tan and Dr. Rob Stadler, who wrote The Stairway to Life, an Origin of Life reality check. And they really talk about all the steps that would need to happen in order for life to emerge. Are you familiar with this book?
Okay, let's be. We don't have much time. Let's
Okay.
try straightforward. First of all, as I mentioned, the scientists disagree with many things. Also, many scientists works in its own scientific model. Meaning, for example, if you're living in RNA world, and for example, if you think that origin of life should be is coming from RNA world, you're thinking about many steps from the perspective of RNA world. When I'm talking about origin of life, my perspective is RNA-peptide world, which is different from RNA world. For me, there is another set of steps which have to happen. Now, also, I have feeling that sometimes creationists try to, because their lack of information, the lack of information of how the science works, what the processes is doing, it is basically immediately filled up with doubt. And the doubt is about there is not possible explanation. None of the scientists, even James, even the Sarah Walker, or any other scientists, you name it, mainstream, it's don't agree that should be uh, some sort of intelligence source for the origin of life, or none of those guys or scientists agree that we exactly must know immediately how it happens. That's the scientific process. Science very frequently surprised us how simple could be something which we thought is very difficult. I can give you a few examples. Two examples I will give you. It's totally different area. You know, I think was in one of the Dead Valley deserts, there were so-called stones that move itself and move itself in the sand and left behind the traces, huge traces. And if you see these pictures of stones that moved, it's kind of it seems that these stones just by themselves, some invisible force moved them on the sand, and it was shock to the scientists immediately how that happened. The stones goes around half a yard away, so how it's possible to give such a nice precise uh, trace of moving over the sand. It sounds like the stone was kind of became life at some point and moved itself there. It happened that explanation is very simple. During the night, there is a thin layer of ice which is covered on the surface, and around morning, this ice is breaking. And if you have at the same moment blowing of the wind, because the thin layer of ice which is covered places a role as a sail, pretty much, and move very easily the stone out. But immediately after 1 hour, all of the ice is just melted away and you have no traces at all. So there is a very clear explanation what happened, although initially was looked completely impossible. So a stone to become life and to move and to do all of these traces. So that's one just example to say that you can have a very simple explanation for something that it might looks like extremely difficult to explain. Another example I'll give you. So the Eastern Islands, So you know that these huge, enormous statues in the Eastern Islands, in an Eastern island, the huge statues that Moa statues that are sitting like that, and there's 10 tons or even more tons of each of the statue, and nobody knew how actually these statues, many statues, are moving. How it's possible to be engraved from the stone and put in the place. There were so many theories, extraterrestrial, some kind of levitation, blah blah, all kinds of ideas, enormously difficult to explain. It was a mystery, 50 years mystery, how it's possible to do and how it's possible to move such a huge statues. Just several years ago, scientists very well showed how this huge status can be moved vertically, just with several ropes and several people, then can handle and move the statues very well, and they clearly demonstrate. This is just two examples that the people and the science and the brave minds can discover how something that looks like completely impossible, actually it's very possible. So
Okay,
about
so
the origin of life, very frequently the creationists put a completely wrong ideas and models in the mouth of the scientists. James Tour is one of those. He presented a picture of a car disassembled of the parts in the beach and said, how it's possible this car to be assembled without any living intelligent designer? Tell me about that. And this is completely wrong because the scientists simply don't have this model in mind. The scientists don't consider steady state situation and never consider that origin of life should be assembly of all of the parts at once. And you have to have, for example, if you think for origin of life as something which you have to assemble very complicated structure, you're wrong. The scientists never think this way.
so you're
In fact,
saying life doesn't require complex interworkings and the chemicals
yes,
coming together.
according to the exactly, I showed you the description of life. The description of life is showing you interaction between information and structure, but it's never show you how complicated it should be. In fact, Terra Darwinia is just one very short peptide or several short peptides and a few short nucleic acid. Terra Darwinia is the first life entity, is only one small RNA with one small peptide, and that's life. Why
Well,
it's
wait a
life?
minute. You said there was no experimental evidence for it. So you're making a very big claim about
Yes,
Terra
I'm making big
Darwinia.
claim,
But
but all of
you
them,
know, in your example about the statues moving, right? There was actually evidence to support it. But in any of your papers, I don't see the scientific evidence to back up the storytelling.
the okay, you should stop telling this storytelling. This is scientific hypothesis.
Okay,
Storytelling
I
is
don't see
something
the
that it's not
scientific
based.
evidence to back up the scientific hypothesis.
Okay, what is the if you can make one yard of walking, can you make 10 yards of walking?
Probably.
Probably, yeah. That's exactly what is happening here. I'm all of the interaction between peptides and RNA are already proven. The aminoacylation processes is already proven. So to combine 1 plus 2 is very easy at that point because you just need another experiment to show everything as a whole how it works. In fact, I'm sure the primitive translation with the primitive codon formation will be demonstrated experimentally. It's completely possible based on everything we know in the science. As I told you, the lack of information in the in the creationist mind very frequently is translated in complete doubt of impossibility, which is not accurate. If you think the first life is a bacteria, the scientists don't think the first life is bacteria. If you think the first life is just Luca containing all of the around 300, for example, genes and interactions, the scientists don't think that is the life formation. Luca is a result of huge and very long process of evolution. In fact, RNA-peptide world says, as a hypothesis, the first life is just short peptide and short RNA. That's it. So thinking from that perspective, life is possible to emerge. But why we call it life in that case? It's very simple, because this structure is capable to evolve. Why it's capable to evolve? Because you have information there. So if I share to you, let's share the screen again. Let's see, is it possible? Share, yeah. So let's share the screen. See, the main problem in many minds, including many scientists, is actually that they think that, in fact, uh, the first life should be in some way more or less complicated. In fact, this is Terra Darwinia. You have a very short, so this is the circles and rhombs which represent the amino acids. You have lines which represent the RNA. You have a process, natural processes, which is cycles, natural cycles like a cooling down, dry and heat cycles, uh, cool and heat cycles, dry and wet cycles. All of this, it's already proven to work exactly in a way that it's done. So here is the formation of so-called hybridization dependent peptides. This is the peptide because you have an interaction, RNA to RNA interaction of aminoacylated RNA. Those process G here and in E is already experimentally proven. I have a paper to show you that. So formation of peptides are completely likely to be expected. Now here,
but where's the formation of these amino acid, where's the all the steps in the process?
okay, but you understand that right now we're in a 40 several minutes of discussion. I can start describing prebiotic conditions and the chemistry part, which already describes the formation of amino acids and RNA. Initially, for example, 15 or 20 years ago, was thinking that RNA is not even possible to happen. But now it's shown that it's possible. If you go in the work of John Sutherland and Dimiter Kunnev and Pauner and many other chemists, you will see that all of these processes for formation of nucleic acids is completely feasible. They experimentally proven. So to go there, I'm starting this hypothesis is just starting from the point that you have short RNA already formed, and of course amino acids, which is already proven to be formed in a prebiotic conditions. Now, the link and to go in a small step from here to formation of actual real information dependent selection is very small. In fact, it's very easy to describe. So you can think about, yeah, it was kind of so simple at the beginning. You said, okay, but now it makes perfect sense because all of next steps are explainable. You can explain formation of ribozymes. You can explain, once you have Darwinian evolution, information dependent selection, you can explain all of next steps. Formation of primitive translation is almost everything in the origin of life. That's clear as that. Now, I want take-home lesson. Take-home lesson. The first life, it's only one small peptide and small RNA interacting each other. This is the first life. Nothing more. It's not rabbits. It's not pink rabbits. It's not snakes. It's not parrots. It's not humans. It is simple peptide with simple RNA. That's how everything started. That's why it's feasible, it's possible, it's statistically relevant. It's also all of the steps are already shown to work. It's simply I didn't combine it because I don't, as I explained initially.
Well, that's the thing, Dr. Kunnev, is that you are not just disagreeing with creationists, you keep picking on them, but what you're saying disagrees with secular.
Yes, I disagree with
work.
many scientists as well. I agree with that.
Okay.
In
So-
fact, if you read my first paper, 2018, I'm against every single scientific view up to date. Every single scientific view up to date, and also agree to some extent to every single scientific view up to date. So there are lots of things that I disagree. In fact, the first paper shows that the idea that you will have self-replication process of RNA, of molecular RNA, is completely wrong view. If someone is telling you that origin of life is self-replication of RNA, I can tell you it is wrong. I do not agree with that. But that doesn't make your case for intelligent design better at all.
Well, I'm not making a case for intelligent design in this on this channel. What I'm doing is trying to examine the claims that are made about the origin of life and the development of life through the process of evolution. So I'm actually not making a claim here. I'm just examining what you're presenting. And here's a very good question I want to ask from Otangelo. It's, how did nature select the 20 amino acids used in life amongst hundreds extant prebiotically? Same with nucleotides. These are very specific complex molecules. Because I feel like you're saying this is a very simple process and you're making life very simple and saying, okay, it was this RNA, peptides, you know, that this was life. But even getting to that point, right, is a very implausible process.
No, it's kind of possible. Let me share my screen. So let's share again the screen. Okay. So here is the view. So I'll answer to Otangelo. Just let's answer it in a slightly longer way. So here is the way from just non-coded hybridization-dependent peptides to coded, the process which I just described. So this is the boundary between chemistry and biology. From that point of view, you started information-dependent selection. And because of this, you select with whatever it is working in terms of making the system more stable and more graduated, the system is more surviving in the conditions. Everything that it's working in this way will be coded because you already have a system of Darwinian evolution. Now, these small peptides, of course, they will start to continue to evolve in a very small motifs. Now, you may have two amino acids next to each other, which create another function. You can put another third amino acid somewhere between those two or around two amino acids further, and you'll have a motif, a small motif which might play a role of facilitating RNA replication, and so on. The several motifs we already know from the approach up, top to the bottom. We have several motifs which is actually very functional. Now, That makes the genetic code formation in a process of formation, meaning recruiting one amino acid after another amino acids. RNA peptide world is talking about the situation where you don't have fully developed proteins, because the protein means folded, long folded peptide, which now adjacent amino acid can get in a close proximity because of the 3D structure. This is not was not possible in a very short peptide. That's why you have selection of the ribozymes, because at that moment you have only short peptides and not much enzymatic function. And now going to directly to answer the Otengel's question about next, how it's possible to come and to select for the next amino acids. Actually, here is what is the revealed analysis. The bridge peptide, which I showed you, which exactly function at the very first interaction between RNA and peptide, actually start to diverse and to recruit more amino acids. Here, for example, is the first set of the amino acids. The first set is glycine, alanine, proline, threonine, and serine. It's coming a little bit later. So this is exactly what you can expect, step by step recruiting more amino acids. But the question here is more important. Why another amino acid should be recruited to the system? The answer is very simple. The next amino acid play a significant role overall the survival of the entire molecular system. So if you have a system which require proline, for example, to make a nick in some place and that to give you another function, those proline will be coded immediately with the available sequences that are available. And so let me, do I have another? There was one very interesting. No, I didn't put it here. Anyway, so I want to show you that if you try to think about the genetic code at the moment is carry on from the proteins, which is called aminoacyl synthetases. This is the enzymes which are capable to recognize at the same time the actually anticodon from the tRNA and the proper amino acid. This is how these particular enzymes, they possess the genetic code in their own structure. And that's according to the RNA peptide world, started with the very short peptide here. Step by step, you form the first class of aminoacyl synthetases, which is class 2, not class 1. Later on, you have development of class 1. And if you try to see how GC-rich codon is in order to the AU-rich, you will see very clearly how this particular classes of aminoacyl synthetases, which can recognize the different amino acids, are very well ordered in zones. I might find the actual picture from the second paper, which is about the codon formation, but it happened immediately simultaneously. If you have a hypothesis which is correct, you can have predictions. And based on these predictions, you can see how the situation is come. Exactly that happened with the zonal distribution of aminoacyl synthetases. Class 2 comes first, and class 1 comes next. And if you make a phylogenetic analysis, you will see that it fits in a group very well. Now, short answer to this long is every new amino acids, it's interacting, it's incorporated in the codon, it starts to be recruited in the new codon because the function fit very well. Now, why this particular 20 amino acids? Because this particular 20 amino acids, it's happened to be useful. That's the answer. The
Okay,
other
so
was not so useful, or there was a selection forces toward having and recruiting whatever it works. You have to understand that the evolution, it's not working in a way that because you have something, possibility immediately will happen. No, it's something that works at the moment and you have is available. If something is available, will be used for evolution. If it's not available, cannot be used for evolution. The same is for the amino acids.
okay, well, you said a lot there. So you're kind of referring to this process 1 and process 2, right? That was in your article, The Point of No Return, The Origin of Life. And so, like on page 10, you said it's hard to explain the formation of system 2 when it is based on prior information and structures of different types, because the selection forces already established number 1 as the well adapted one, right? You're like kind of discussing this difficulty of, hey, we need this.
No, let me describe what I'm discussing. There are lots of hypotheses, like, for example, RNA world, which says you will have RNA entity. And this RNA entity, it's already selected to exist because this RNA entity have self-replication system of the RNA because of the ribozymes that is doing this. But suddenly, after this entity is already appeared, RNA, this entity is based on information from RNA and the structure also from RNA. So you have RNA information and structure. What the RNA world supposed to describe, that once you have RNA world entity, you have to have a polymer transition toward RNA protein world. And this, it's need explanation why you should have this particular transition from RNA only entity to RNA protein world. Why? Since you already have established selection toward most selected RNA entity. The reason to going from RNA only to RNA protein world should be some sort of another selection based on the first one, or information description based on the first one. But you cannot leave behind one of the selection and information and take another one on the top of that. It simply don't work in this way. You must have something which benefit the next step. You cannot just throw away. If you start to build up a house, you cannot start building the roof when you remove the basement of the house. You don't do that. This is all about in this page 10. It's a description of, if you wanted to work on the origin of life, you should work on something which already started in a system which we already know. And this is the translational process. Did I describe well?
Well, I mean, I hear what you're saying, Dimiter, but you've said that you're contradicting what many you said all
Yes,
origin of life researchers are saying. Okay.
Pretty much
And
all was,
you're making it sound like this is a very simple process and that we've shown the scientific processes by which this can happen. And I haven't actually seen that in any of your papers, and I haven't seen that demonstrated by other origin of life researchers. I have Dr. Stadler and Dr. Change-Loratan saying that there's, all these steps that would need to happen. And we've got, formation. First, you've got to form the 20 amino acids, 5 nucleotides. Then you have the homochirality problem. Then you have the paradox of water that requires, you need water, but it's also destructive to life. You've got to have that. You have this homeo linkage problem. And I don't understand all of this. And this is why Casey Randy says this is over most people's heads from the beginning. And that's true. When I read this, when I read your papers, I don't understand all of the scientific details, but what I do understand is that you're proposing a scenario that has not been experimentally shown to be viable. And I'm wondering, why should I accept your proposal? What's the scientific reason I should accept your proposal that contradicts our scientific observations?
Does not contradict scientific observation. Why you think my proposal contradicts scientific observations? It contradicts another thinking and another scientist. That's what contradicts. It does not contradict scientific observations.
Okay, let me ask it in a different way. What are the scientific observations that back up what your proposed hypothesis is? Where is the scientific observation? What's the experiment or set of experiments?
Which is already published in the papers about aminoacylation, for example, of short RNA. It's already published. There is experiments which shows that it's possible. The second hybridization process of RNA, I don't need even to try to prove anything to you. Everybody is doing PCR now. PCR is completely viable experiment. Every day, thousands of PCRs are done, especially now with COVID. The PCR process is exactly that, hybridization between nucleic acid. So you have hybridization process and cycle of the hybridization process. So I don't need to prove that at all. I mean, if someone tell me that this is not possible, I mean, is it completely ignorant or what? I mean, now, having in mind that hybridization process is not even slight issue, having in mind that the aminoacylation process is not issue at all, of course, we need to figure it out, conditions and so on, but it's feasible. And having in mind that you have interaction between positively charged amino acid with negatively charged RNA, I mean, are you doubting that plus will interact with minus in a charge? Are you doubting that? I mean, come on. That's not even possible to doubt. There is no even.
What I'm doubting
I will accept argument against this particular hypothesis if you take every single step and say, put your finger there and said, scientifically, this is impossible to happen because of something which I could agree might not happen. So far, I have at least maybe 10 reviewers in the papers, this particular. So if you summarize all of these papers, I have at least 10 reviewers over in these papers. Nobody said that you will be stopped in this particular point because that's not possible. Not to mention that all of these papers has been already cited in one way or another, and there was not even single criticism that some of those steps might not happen because of something. As I said, every single step is scientifically or experimentally proven steps. So you cannot argue against the fact that you have highly plausible scenario. That's it. And the fact that it's highly plausible eliminates my need to use intelligent designer. I don't need intelligent designer to prove the complex between RNA and peptide, and also to prove this information-dependent selection. I don't need intelligent designer to show me, to demonstrate in information-dependent selection in action. That's even not issue to me.
What's your response to Mr. Jetty 89? PCR can only happen in a test tube, clean, very clean environment.
That's not even correct.
Okay.
I
Tell
mean,
us why.
you can put in the water RNA. Of course, there is a degradation processes, but that doesn't mean you cannot hybridize something in the pond.
Okay. What about this, thing that is mentioned? I think it's called like the asphalt problem, where,
What kind of problem?
the asphalt problem,
Asphalt.
where you've got, you know, the tendency toward deterioration. And when you've got these chemicals working together, they don't produce anything. Like when we observe how they react, it ends up with just a mess. It
So,
doesn't order itself.
because we don't have much time, I will be kind of just describing the situation. But for someone that it's interesting more on this, let's go to the John Sutherland and see the entire prebiotic chemistry of the process. Dimiter Kunnev also is a guy that describes how UV can go and produce toward specific nucleotides. Also, there is a very cool experiment from from the Jacques Zostak about to say that if you have different types of nucleotides, after all, the most stable is the RNA that we know it. And now coming to the James Tour, which assuming that everything is steady state, the beach is there and the car is disassembled and nothing will happen. We don't work in this type of chemistry. The chemistry is a floating process. You have cycles of different heat, cool cycles. You have cycles between dry and wet cycles as well. You have all kinds of processes. You have a pipeline from the streams, pipeline which can go from up to the bottom. There is a chemistry which is going under the earth, from the volcanoes, from the UV, all kinds of stuff. It's to say that chemistry stay and they don't produce anything, it's completely ignorant. I mean, we don't want, we you don't take a cope with some kind of reaction inside the cope and try to make a life there. That won't happen. This is a cycle. You have dynamics, constant dynamic and selection.
Well, I think we're going to start wrapping it up now. If anybody has any burning questions, please do put them in the chat right now. So, Dr. Kunnev, is there anything that you feel like you've left out that you wanted to mention?
Well, it's very important to take home lesson is that the first life should not be complicated life. It's not necessary to be complicated. The very first life, which is Terra Darwinia, is just a very simple peptide with RNA capable of Darwinian evolution. That's it. It's simple as that. And how stable is? Just because it's more stable than everything else around, that's why it's existed. The main forces is the stability. And the reason why you have codon formation is because this particular sequence of 2 or 3 or 4 not amino acids, but nucleotides with interaction with this particular peptide makes the system more stable, slightly more stable. And this is information by itself. So information, it's created right now, as I told you, it's very simple. Just because you have one sequence of RNA, which makes the sequence of the peptide, it's already information. So, and the second thing that I want to mention, Discovery Institute misrepresent the scientists, misrepresents the models we scientists working with. If you say that the first life is just ready bacteria, that's a wrong misrepresentation. We do not think in this way. If you say that the life you have to smash all of the peptides, all of the proteins together and make something very big, it's a wrong thinking. We scientists do not think this is correct way of thinking. There is no even single mainstream scientist which says, oh, life should be created with peptides immediately. There is no such, meaning proteins. Life cannot make proteins. The proteins, life makes proteins, but meaning the origin of life is not immediately emerging of proteins. There is an evolution of the proteins. You start with very short peptide. After that, you have motif. This motif make a domain. Why? Because now you have 3D structure. And something adjacent can go closer together. Now, because you have 3D structure, you have domain formation. This domain is doing something. And finally, you have a protein which is long and contains 1, 2, or 3 domains. That's now we have a modern protein. So, but that doesn't mean the protein it's appeared out of the blue. Because first of all, it's not possible. And all of the creationists explore the possibility for making protein. Of course, it's not possible. Nobody disagree with that. But we don't work in this model. There is no such a model in origin of life at all. So making a wrong models and fool the people that is listening is the way how Discovery Institute works. The same is true from James Thorpe. The same is true from Stephen Mayer. The same is true for everyone. Even X was presented completely wrong concept for how the evolution works. It's a wrong concept. The evolution does not work in this way how they present it. And of course, it's not possible because the model is wrong.
Okay, well, thank you for expressing that. And I will just, in response, you've presented that Terra Darwinia was the beginning of life, and you've described this, with the short RNA peptide explanation. But by your own admission in your paper on page 14, the very first steps toward the formation of primitive translation and primitive enzymatic replication are not experimentally demonstrated yet. And so, there's a chicken and egg problem that you refer to quite often here, and that has not been overcome. And it's not me as a layperson saying this. I don't even understand all of the stuff that you guys are talking about, but I'm basing this not only on what the intelligent design movement scientists are saying, but also on what the secular researchers in the origin of life are saying, which is that this process by which non-living matter could become living and all the steps involved is one of the greatest outstanding problems of science. These are secular researchers saying this, not creation scientists. And they're saying what we're looking for is a way to overcome the laws of physics as we understand them in order to get the origin of life to happen, because that is what is needed. So you're presenting this as a simple process that, it's easy, we can demonstrate it, when I think that is a complete misrepresentation of the complex and extremely difficult, and I want to say impossible processes that would have to happen in order for the origin of life to occur.
So let me finish. First of all, RNA-peptide work completely solve the problem with chicken and egg problem. Chicken and egg problem is completely solved exactly by RNA peptide work. There is no such a problem at all. If you read carefully the actual paper, you'll see that. The second, lack of information usually is translated in a doubt. And that's not a good idea. Just go and look. There's nothing mystical about having interactions between RNA and peptides. There's no mystical there. Also, the third thing, how complicated is life? Of course, it's very complicated. The life is super complicated now. That doesn't mean the first life was complicated. It's simply you have a process of evolution. That's how the things are going. And the fourth thing, you might think that the life is super complicated, but in fact, it's created just from several blocks. All of life forms are created from 20 amino acids and codon formation, and everything is just recombination, making something better, like a Lego game with the blocks. You can make a castle, but you can make a car, you can make trees, whatever. You can make house from the same blocks. That's how the life actually is. You have a very simple beginning, and now you have more complicated. And the proof for this is, of course, exactly the prediction I mentioned. If you're thinking this way and you can go backward, you predict what you have to find. You find exactly that in the genetics, in the phylogenetics tree, in the fossil records, everywhere. So we'll be surprised, I guarantee you. Very soon you'll be surprised.
Okay, well, thank you so much for your time today, Dr. Kunnev. Thank you, everyone, for watching, and have a great day.